Good day. I'm Kristen Hunter, Marketing Communications Manager at Pannar Seed. Today I have with me Rikus Kloppers, who is an integrated field science leader for the Africa Middle East for Corteva Agriscience, and Nico Barnard, Pannar Seed's agronomist for the Gauteng, Southern Mpumalanga, and Eastern Free State region. Together they co-authored this presentation. Rikus is a well-known figure in the South African agricultural community. He has over 32 years of agricultural research experience and has authored and co-authored many publications and made scientific contributions at congresses and workshops around the globe. We are honored to have him present the third and final presentation in Pannar's NAMPO virtual series. The topic today is Sclerotinia, understanding and managing this disease in row crops. Welcome, Rikus.
Good morning, Kristen, and thank you for the kind introduction. Thank you to everybody for attending this morning's session on Sclerotinia, making time for this. And for the sake of just a proper internet connection, I am going to shut down my video now and just talk behind the slides. So thank you to that. And also just want to acknowledge my co-author on this presentation, Nico Barnard, who's also a well-known agronomist in the field of soybeans. So thanking him for his contribution. So yes, let's start with this NAMPO virtual session on Sclerotinia and talking about managing the diseases in row crops. And if in the two row crops that I'm really going to focus on today is sunflower and soybeans, although those are not the only crops that Sclerotinia can attack. There are many others as well. But let's, for the sake of this, focus on these two. So I think very sad that we cannot be in Botawil at the NAMPO exhibition this year. It is such a big tradition and such a nice place to be, to go to the stand there and meet the team and just looking at the wonderful exhibitions and meet so many people and stakeholders in agriculture. But unfortunately, we have to do what we have to do this year and make the best of it. So the technology helps us in certain aspects so that we can at least still bring a message to you. But on the other hand, we will be missing out on some of the things that NAMPO always brings us as well, and that we will have to forfeit this year. But anyway, let's get back to the topic, as I say, talking about Sclerotinia in the two crops, soybean and sunflower. So Sclerotinia is caused by a fungus called Sclerotinia sclerotiorum. And there's, and just looking at the symptoms quickly, this is very typical of what you see in the soybean, is that white mycelium growth on the stems around where the flower used to form, and then eventually that white mycelium forms a hard structure called the sclerotia, which is the survival structure of this pathogen and very important part in the life cycle of this pathogen. If you're looking at the field symptoms, so there's two pictures here. The one on the left, the green field, a picture that I took in the Greytown area, you can see all those dead plants then evenly distributed all over the field. And that is usually the first indication normally that farmers see when they find Sclerotinia in the soybean fields, that suddenly these plants are starting to die. These individual plants all over the plant, you see them wilt and die. And then when you open that canopy, you see that white growth of the Sclerotinia. So you can see that distribution. In the big picture, behind this, what you see is a very localized area in a field of soybeans. Also a picture that I took this year in the Standerton area, which is very symptomatic, again, of a very small microclimate that favored the disease development. So a small patch in the field where there was obviously enough moisture around, so the sclerotia germinated and infected that area. And you can see the rest of the soybean field up there is actually perfectly healthy. So it can also be in these hot spots. In sunflower, we get different types of symptoms. The one is the Sclerotinia head rot. As you can see on the slide, on the back of the sunflower head, and later on in the season when the plant dries down, you will find the sclerotia in those dead sunflower heads. What is very typical of the Sclerotinia head rot symptoms is wherever you see the symptom on the back of the sunflower head, if you turn that flower around, you will also see the white mycelium growth in the front, which makes it a bit different from other head diseases like botrytis and rhizopus, where you normally just find the back of the head rotting and you see the gray mycelium. And this is always on both sides. And this white mycelium in the front eventually then goes black and forms the sclerotia, and it can even form a nice, like a honeycomb structure of black sclerotia that eventually end up falling on the ground in a field that was, where you can see there was a very serious infection on almost every sunflower. It was infected, it becomes stringy, the seeds just drop out, and the sclerotia with that all end up in the soil. The other typical symptom of Sclerotinia on sunflower is also this basal stem rot or wilt symptom that you see there. And what is very typical with this is where you will go to a sunflower field or planting and you will see suddenly individual plants here and there suddenly wilting, and it's almost like it's happening overnight. You can see this plant is still green, but it's just wilted. And then normally what is a giveaway that it's Sclerotinia, if you go to the base of this plant and you go down, look at it, you will find this white growth of mycelium on the base, which is very, very symptomatic, typical of Sclerotinia. But you can see also there's some of the other plants standing right next to it, which is not affected, and I will explain the reason for that coming to the life cycle of this pathogen. So I think whenever we talk about a disease, the most critical thing is how do we control this disease? How do we manage this risk? And Sclerotinia is definitely one of the most difficult diseases to manage, to control, and definitely my key message today is there's absolutely no silver bullet to control this disease. And to be able to understand the disease and how to manage it properly, I think we have to go back to the basics again, and I think most of you have listened to my previous talks. I will never go into a plant disease talk without mentioning the disease triangle, because I think that's key, and especially key in a disease like Sclerotinia. So what is in the disease triangle? There's three aspects there. There have to be a susceptible host, the environment have to be conducive for the disease, and there have to be a pathogen present. And only once you have all three of those components in a specific growth season, in other words, in time, I put it in this box, and then you will get disease. So if you take any one of those components out of the disease triangle, you will not get this disease. And in Sclerotinia, I think we know the susceptible host, we know sunflower, we know Sclerotinia, but there are also many other hosts. If you look at Sclerotinia hosts, they were saying there are almost something like 64 plant families that can be infected by Sclerotinia, and around 400 or so different plant species, mostly your broadleaf type of plants. But here is also a picture showing a cabbage that has been infected by Sclerotinia, but there are many other weeds and potatoes, and just too many examples to mention. So that is the host. The pathogen itself is the Sclerotinia. As I say, the sclerotia is, I think, in every hectare in this country where sunflower or soybeans or any other broadleaf have been grown over the last 50 years. Definitely the pathogen is all over. The key one to me is still the environment in Sclerotinia, and it's critical. And that is why some years, there could be many seasons that you go without seeing any disease, and suddenly it pops up in a land where you didn't have any disease, and you would think, where does this disease come from? And it's really all about this critical aspect of the environment, and matching 100% the susceptible host and the presence of the disease in the soil. And we can see critical aspects is the soil moisture for 10 to 14 days, from which the apothecia can develop from the sclerotia. And you can see also we need lower temperatures and leaf wetness so that the ascospores can infect the flowers or the stems wherever. And as soon as you go higher temperatures, the disease is really suppressed. And I've often seen that you almost need a bit of a cold spell to initiate the disease together with that humidity or moisture that's around. And once you understand the disease triangle, I think then you can start thinking about how you can manage the disease or the disease risk. So to me, that is key. Really, again, knowing your enemy is the first step in the management strategy, very important in the life cycle. And this slide looks very complicated, but I can really assure you it's not that complicated. There are two parts of this life cycle of Sclerotinia that you should recognize. There's a blue, there's a red life cycle and a blue life cycle. And the red lives, and it all really starts with these black parts here with the sclerotia produced in the disease plants, and the sclerotia then sitting in the soil, and it can survive for a very long time depending on the condition. And in the red cycle, you really get that sclerotia that infects the roots very close up. The plant wilt and die, the fungus grows in that plant, sclerotia is produced, and it survives. And that is the cycle of this myceliogenic germination part. The other one is the blue line that you follow here, and that is very critical, especially in the head rods of your sunflower and most of your Sclerotinia in soybeans that you will find. And that is really the part where the sclerotia sits in the soil under good conditions. These sclerotia germinate, they form apothecia, and within those apothecia, they produce asci, or really bags with spores in it. The spores are released and transported by the wind. These spores fall on the middle stalks and on the flowers of the plant, infect the plant, and eventually you go back to the sclerotia in that cycle. So that is the two cycles, and I've actually just broken them up into two cycles with pictures. And here you can see in the red cycle, the myceliogenic germination, the sclerotia, that black structure germinates, and what happens here is where there's a root growing very close to that sclerotium, the sclerotium germinates and infect that root and that base of the plant directly. Infect, you see that wilting symptoms, and the plant die and produce new sclerotia. The blue cycle is the more complex part of it, and the carpogenic germination side is the stem and the head rod side. As I say, again, the sclerotia sits in the soil, can survive for a long time, it germinates, and then it forms these little cup structures called apothecia, and in that apothecia, it forms asci with ascospores. There you can see an ascus with ascospores. There's normally 8 spores in such an ascus. They get shot out, and in that picture you can also see it. And this really happens after, when the soil moisture is there for 10 to 14 days. Then these apothecia develop, and the ascospores are released. They infect the middle stalks and causes the symptoms that you see on the sunflower, on the head, or on the stalks of the sunflower, and you will see it in the field. So this is again just emphasizing the importance of that environment and the growth stage, very specifically on the soybean between R1 and R2 reproductive stages, so that early flower formations, lower temperatures, wetness periods, and dew or mist or something that causes the disease, the infection to happen. And also just mentioning up here is how the disease then spreads up the plant. And normally about 3 weeks after flowering in the soybean, you will start seeing the dead plants appearing all over the field. And that is the percentage of plants that will be infected. So this disease doesn't spread from one plant to another. It actually infects that plant when the spores are released. When there's a specific place where that spore then lands and the environment is suitable, that plant will be affected, and it doesn't mean the next plant next to it will be infected. So it doesn't spread from one plant to the other. And that's why I mentioned that we are monocyclic versus polycyclic. So polycyclic diseases is something like rust, where there's one spore that ends up on a plant, forms a rust pustule, produces new spores, and within the same season, they will they keep on infecting. In monocyclic, that infection happens once when the conditions are favorable, that plant gets infected, and and then only the next infection will happen the next year when the cycle repeats itself. So that is just going through the life cycle quickly. And then the other thing that we often find is just that there's still some confusion about the apothecia, what it looks like. On the left, you see the apothecia that is formed by Sclerotinia sclerotiorum, and these releases the spores. So there you can see the sclerotium, the apothecia forming out of that. You can see the size of that. On the right-hand side, you see what we call the bird's nest. It's a friendly fungus. It is responsible for breaking down plant material, organic material. And it's often confused, or people would think that this is a Sclerotinia. And once they see this, they start spraying chemicals. So just be very careful and make sure that you can be able to distinguish between the two. In terms of disease management of the disease, there's lots of practices. As I said, there's no silver bullet, and that's why I've lined up quite a few bullets there. But really, if you look at it, all of them using quality clean seed. A lot of agronomic practices that you can follow, like your water management, crop rotation, weed control, tillage system, your canopy management, in other words, row spacing, things like that. Preventing combines from moving the sclerotia around. Then hybrid or variety selection, depending on what crop, using planting dates. And then we come down to chemical control, biological control. And if you combine a lot of these strategies together, we are referring to that as integrated approach. So if we just look at some of them individually, I think critical to start off, you have to start with good seed. Quality seed, clean seed, healthy seed is very important. So in the seed industry, there is certain rules that have to be followed. So in soybeans, when seed are sold, there's 0% sclerotia tolerated in the soybean seed. And you can see why, because it's really very easy to see the sclerotia in the soybean seed. You can easily pick them out using the proper equipment like gravitation tables, etc. And then obviously also combining that with an appropriate seed treatment that controls the disease can also assist. In sunflower, there's a tolerance of 0.2% of sclerotia because it is slightly more difficult to remove the smallest pieces of the sclerotia that can be stuck somewhere in the seed. And again, that is where there's an appropriate seed treatment applied, which will control the disease or at least suppress the disease, whether it's internally or on the surface of that seed. So start off on the right foot is key. Some of the agronomic practices I've mentioned, I think it's some of them is very logical. As I say, once you have a disease field and you move from one field to the next, you can move sclerotia around and really in that way spread the disease all over the farm. So try to minimize that. Using proper water management, looking at those critical stages, those reproductive stages, so that you do not overwater and create an environment suitable for infection. I was mentioning weed control. So many hosts and so many weeds can also be infected by Sclerotinia, like you can see here, the cocklebur down here, also infected with Sclerotinia. So weeds act as hosts for Sclerotinia as well, not only the crops. And then working with crop rotation now, which makes it very difficult that there's so many alternate hosts in this. So you can't obviously rotate now soybeans and sunflower. That's a serious issue. But because the sclerotia survives in the soil for so long, you really can't go to a crop rotation of a 5-7 years a rotation before you go back to that crop. And that makes it very difficult. But at least with crop rotation, you can reduce the number of sclerotia that ends up in the soil. So it is part of the management strategy, not the ultimate control system. Tillage and fungal survival. You know, with tillage, if you start burying the sclerotia very deep in the soil, they would actually just survive for much longer. While if they are on the top of the surface below 5 centimeters on the soil, they will germinate and form new apothecia and release spores. But if you have those sclerotia germinating and there's no host that they can infect, for other words, wheat or maize, one of those monocot crops, the spores gets released and the sclerotium really gets destroyed, but there's no host that it can infect. So in that way, the sclerotia on the top actually gets destroyed as well, or reduced in numbers. I always say, you know, when I look at this picture of the frost on the plant, we must remember that cold doesn't kill these fungi. They would actually just survive much longer. So don't think if we have a very cold winter that we will get rid of the diseases. The other one in agronomy is just row spacing. I know we're looking at 91, 76, 45 centimeters. And again, here it's very important to realize that the higher your plant population and you have canopy formation, you do create an ideal microclimate for disease development and can create a lot of disease problems. So going wider rows, and I think practices of your more than 1 meter, 1.1 meter, then you start opening up between the rows and you don't have that canopy formation and the microclimate. But very important to realize that there will be a trade-off for yield when you start going with the lower plant populations. And then varieties, a lot of questions about that. Screening a lot of sunflowers and soybeans over the years, and there's very small differences. I can assure you, very limited success. We have seen very subtle differences between varieties. And I often think when we see two varieties next to each other, where the one gets disease and the other one not, it's more often a case of escaping the disease, where the one is just not flowered yet and the other one was slightly earlier or later, that you really escape the disease because the spore lands somewhere where it cannot infect. And also just looking at the use of different varieties, as Nico pointed out here, if you look at those quicker varieties, you know, the days to flower, 49 to 52 days. So start that season with the earlier varieties and you can escape the disease. And you can see that then there's a gap between the 52 and the 57 days. And again, with a very long growing season hybrids, you can see 63 to 67. So playing around with those flowering dates, because that's where the infection takes place at flowering. So you can break that cycle so that you do not have a continuous flowering and infection that takes place. Biological control, there's a success story in biological control, and many years, and I think in the early 90s, I worked with contents. It's a fungus, Corteva militans, which is a fungal pathogen that parasitizes Sclerotinia. You can see the pictures. These I actually took there at the Dalma site. You can see the sclerotia, where you apply the contents on the soil. It finds that sclerotia in the soil, attacks it, and really destroys it. So in that way, at least you can reduce the inoculum pressure right at the beginning. Chemical control, as I say, a very important one in this whole group. But key again is timing and adequate coverage is the two most important components. The timing must be spot out when those ascospores are released and when the weather is good for the disease. And the other thing is also penetrating that canopy to get the proper coverage. So as I say, not 100% success with chemical control, but definitely there's a success story in this. But if you understand this disease cycle, as I mentioned earlier, and you target a pathogen at its most sensitive state, you can get a decent control. But it really means looking at things like increasing your spray volume, keeping your ground speeds low, and then, as I say, using proper nozzles that can penetrate that canopy to at least the middle of that soybean plant so that you can reach the point of infection or where those ascospores then land on the plant. And soybeans, as I said, I mentioned earlier, that's really, you can look at the early reproductive stages where you need to do that first application and reading the labels of some of the fungicides. It could be there, and then you might have to repeat it for up to 3 times during that same season. In sunflower, I think the window for the infection is slightly bigger. You can see that early infection, and sometimes you might have to go with a preventative spray, but later on you have to act preventative and more curative, but as I say, the window of infection is slightly more, but it also happens during the reproductive stages where you have to target the pathogen. There's actually only one fungicide that is registered in South Africa, and that is the active ingredient prosimidone. And in the Corteva group, we have registered that product. It's called HEAT 500 SC, and you can see it does have some curative action because there is a bit of a translaminar local systemic in the leaf, so it does spread a bit. It's rain fastness, but if you, as I say, with fungicide, be very important to follow the clear instructions on the label. You can see in dry beans and soybeans, even up to 3 applications of the same of the product at 10-day intervals to get some kind of control. Then just to take all of this and bring it together, as I say, if you look at this life cycle of the pathogens, you can see, and I've tried to indicate that the different areas where you have to do something to manage the disease. And if I start here at the bottom, you know, with your sclerotia, use your biological control, with the proper tillage, or those type of things. When the epithelium releases here at the reproductive stages, do the scouting, look at your weather, and if the environment is conducive to disease, that's when you apply your fungicides at that stage. When you look at your crop, your proper crop rotation, so in other words, I was saying the sunflower, soybeans, and the broadleaves do get the Sclerotinia, maize and wheat will not get it. So pick your crop, do your crop rotation where possible, use the quality seed, use seed treatment, proper row spacing, variety selection, all of those things can add to the management strategy. And then eventually, if the plants that the disease develops, do not spread it around with equipment from on your farm or from the one farm to the next. So with that, I would end here. As I say, key messages: no silver bullet to control Sclerotinia. It really takes an integrated approach, and understand the enemy, understand this pathogen, because then you can manage it properly. And to me, very critical in this disease is the environment. So we know biology loads the gun. The pathogen is out there in the soil and in the environment, but the environment really pulls the trigger whether we're going to see disease or not. So that's all from our side. Thank you very much for your attention, and we'll be taking some questions at a later stage. Thank you, Kristen.
Thank you, Rikus. Thank you for your time and a very valuable presentation. As Rikus said, you can direct your questions to us by email, or we are still available at the NAMPO stall until Saturday, 12 o'clock. Thank you, Rikus. Bye.